Hi, I'm Richard, one of the imaging scientists at IXICO. I'm going to use this short slide deck to do a whistle-stop tour of an MRI technique that we're particularly interested in at IXICO, something called free water imaging. I'll very quickly summarise what this is and how it's useful, using the study of Alzheimer's disease as a working example. So, when we talk about free water imaging, we're not actually talking about a novel MRI sequence. Free water imaging, or free water correction as it's alternatively referred to, is a post-processing technique that you can apply to an already well-established type of scan called a diffusion MRI. So I'll start by doing a very quick primer on diffusion MRI. So diffusion MRI is as I said, it's a well-established technique. It's been used to produce hundreds of journal publications in neurological and psychiatric conditions over the past 20 or so years, because these sequences can reveal various interesting properties of the tissue that you can't easily see with other types of structural scan. The way they do this is by using a series of very powerful magnetic field gradients to track the motion of water through the brain, and then the interaction of water molecules with the surrounding tissue gives us clues as to the brain's structural organization. Now, something that's often pointed out is that diffusion MRI is it's a wonderfully sensitive technique, but it's not overly specific. You may well find notable changes in brain diffusion between experimental groups, but without strong prior knowledge it's very difficult to pinpoint exactly what biological process could be driving that change. So if you think about the millimeter scale resolution of a clinical scan, each let's say a 2 millimeter cube imaging voxel is actually summarizing a huge and varied cellular landscape. And for one pertinent example, what we end up doing is mixing signals from the water that resides within cells, the intracellular space, with the water that sits in the interstitial extracellular space, and this isn't ideal. So this is where free water imaging comes in. This technique uses some prior simplifying assumptions about the characteristics of unhindered, unrestricted water to help us split apart the unique contributions of intra and extracellular water to the diffusion signal, so we can analyse these contributions separately. In a nutshell, the technique relies on something called a bitensor model, where you're fitting two tensors, one per tissue compartment, that adequately explain the measured signal, and we do this on a voxel by voxel basis. Okay, so we'll now briefly consider Alzheimer's disease and AD clinical trials. So there's currently a lot of interest in the free water technique for the study of Alzheimer's, given the recent interest in neuroinflammatory mechanisms underlying this disease. So we now know that there are a few of the hallmark proteins involved in AD, including beta amyloid and tau. They set off inflammatory alarm bells within the brain, leading to an immune response that ultimately leads to cellular damage and death. As a consequence of this, several trials are now focusing on compounds that target this inflammatory chain reaction within the brain. Importantly, these kind of inflammatory processes have a much greater impact upon the extracellular compartments, and so it's hoped that the free water imaging technique may give us a more precise view of this particular dimension of Alzheimer's disease. IXICO will be incorporating a free water correction approach into our diffusion MR analysis pipeline this year, and we hope to have some interesting results to share very soon.